first, we're going to be presenting a brief presentation to give you all an introduction about Sharjah Research Technology Park. I think my screen now is visible. So Sharjah Research Technology Park was founded back in 2016. His Highness invests annually millions of dollars that goes to education. He has paid a lot of attention and a lot of funds that go straight to the education system here in Sharjah. We have many great universities that are heavily focused on research and development. And the Science Park was founded to commercialize the research, which is based in the universities, and bring it to the market. At Sharjah RTI Park, we focus on four main categories. We are building an innovation ecosystem by supporting our partners to scale and grow. We are generating employment by developing talents and entrepreneurs. We are catalyzing knowledge economy and investing for the future. So Sharjah RTI Park is a science park, and we're also a free zone. We offer different things to startups, SMEs, corporates. We work closely with governments, venture capitals, and accelerators and incubators as well. And the services provided for each of the above is different. So here the slide presents the benefits of establishing at SRTI Park. We have 6 anchored deep tech projects focusing on different technologies. For example, we have photovoltaic solar panel systems. We have agriculture technologies. We also focus on 3D printing in 3 main areas. We are the only place in the Middle East that combine metal printing, plastic printing, and also a lot of other places as well. We also have an active manufacturing hub, AR and VR experimental center, and mass transport innovation center here. We have over 80 companies registered here at the park. And we focus on 6 main areas: environmental technologies and renewable energy, digitization, mobility, logistics, and smart cities, production, design, and architecture, water technologies. We have launched our first advanced accelerator program back in 2020. We focused on main topics such as additive manufacturing, modularization, smart construction. We have successfully received over 500 applications from 48 countries, and we're currently working with startups that have won the accelerator in order to help them grow and advance in the market. So I'm going to give the mic to Dr. Jalal Tanira.
Thank you very much. Thank you for the presentation, Hamda. I'm going to share my slide now. Okay. Okay. Yep. So first of all, thanks for the invitation. I'm really glad to be with you and talking about my work. My name, of course, is Jalal Tanira, and I am assistant professor at the College of Medicine, Sharjah University. And my talk will be about the identification of novel genes that involve in insulin secretion and type 2 diabetes. And I will take GPR 183 gene as an example for this talk. So to start up, just a few words about type 2 diabetes. We can say it's a complex and multifactorial disease that ascribe for an interaction between common genetic variants and environmental factors such as obesity. The type 2 account for more than 90% of all diabetes cases worldwide. So far, Genetic Wild Association, a study have identified more than 140 genetic variants that associated with the disease. However, these 140 explain only probably between 10 to 15% of the heritability of the disease. And in most cases, these genetic variants, it's not really clear how they influence the islets function or how they involve in the pathophysiology of the disease. So based on that, this has really provoked scientists all over the world to develop a new strategies to identify putative or novel genes for type 2 diabetes. However, this has been all the time difficult because I would say 10 years ago, we did not have an access for human pancreatic iris, which is really a big obstacle when it comes to diabetes research. So we were fortunate, I would say, 10 years or 9 years ago, when I was working in Sweden at that time, to start building up a collaboration between Lund University and something we call it the Nordic Network for Iris Transplantation, where they start to provide us with the human pancreatic iris. We were able to build up really a good, one of the biggest cohort when it comes to the human pancreatic islets. So at that time, we were able to collect almost like 200 islets from 200 donors. Sorry, we collected islets from 200 donors. And these islets, of course, must be really characterized before doing anything. For example, we take, for example, some of these biomedical information about age, BMI, hemoglobin A1C, gender, insulin secretion, and etc. As you can see, this is on your left hand or on your right hand, I don't know. You can see this is one of the human pancreas that we used to get from, of course, from dead cadaver donor. And we start to isolate, which is a very complicated process, to isolate the human islets that you can see it here. So these human islets that are responsible for the disease represent all with, by max 1 to 2% of the total volume of the pancreas. And here you can see a kind of immune staining for all the type of the cells in the human pancreatic iris, where beta cells represent 70%, alpha cell represent 20, delta 10, pp5, and green cells present 2%. So these iris, we used to take them and subject them to RNA sequencing or a microarray. We started first at that time with the microarray, and then we moved to the RNA sequencing. And we were able to combine data from all from the human islets gene expression, in addition to the genetics and function, to build up a global map for the genes that associated with the islets dysfunction and type 2 diabetes. The roadmap that we used to do at that time, of course, is to compare the gene expression between diabetic islets and non-diabetic islets. Doing this correlation, we end up with some genes, of course, many genes that could be differentially expressed by doing such analysis. Of course, you need to end up with a sort of ranking. I mean, what is the important and what is the least important genes that you need to follow up and to validate. So to rank the gene and give them a priority, we used to have 5 different criteria. For example, whether this gene has any kind of genetic variants that involve with the disease, whether this gene differentially express between diabetic and non-diabetic when it comes to beta cells, or when it comes to the islets, and then whether this gene correlates significantly with the hemoglobin A1C or insulin secretion. So based on these criteria, we used to rank the genes from 1 to 100. And of course, the less rank, the better chance that we need to validate this gene. So I would say doing this and having a list of these genes, we comes to the functional validation that we use, of course, to do in rat NS1 cell, which is one of the best model for translation research to diabetes in our community. What we used to do is to silence the target genes by sRNA and then try to follow how the silencing of this gene affect the beta cell function when it comes to when it comes to the insulin secretion, for example, when it comes to the insulin production, when it comes to key pancreatic genes that involve in insulin secretion. So this is like the main road that we used to do. Doing this, we were able to publish several papers about novel genes for type 2 diabetes. And we're still in process to publish more and more using this approach. So this is the first part, but I'm going to talk about another specific talk today. It's about the G protein coupled receptor family. As you know, this is the largest family of integral membrane protein involved in microbiological process and pathologies. Remember that mostly 50% of all modern drugs and 25 of the top 200 best-selling drugs are estimated to target the G protein coupled receptor. They transduce, of course, a signal mediated by diverse signaling molecules such as ion, peptide, lipids, photon to induce different intracellular function. And they bind, of course, the ligand to activate different G protein. If you look at the literature, there are more than 700 G protein coupled receptors that have been identified. However, 100 of these G protein coupled receptors, they lack defined endogenous ligand. Thereby, we call them orphan receptor. These orphan receptors, their role, expression, implication, insulin secretion, or in human pancreatic islets are poorly understood. As well as a study on uncharacterized orphan receptor are of great importance to expand the therapeutic target for diabetes. If you look at the table here, you will see 4 different drugs that have been, or they are under development now, that based on the G protein coupled receptor. We have G protein 40, GPR 190, GLP receptor and glucose receptor, GLP receptor and GIP receptor. So these drug under development for type 2 diabetes. So we thought maybe we can expand this list by looking for another target using these 100 orphan receptor from human pancreatic islets. So what we have done here, we used the International Union of Basic and Clinical Pharmacology database to list all the orphan receptors. We end up with 91 orphan receptors. So thereby we use the microarray expression and look at their expression profile in human pancreatic islets. By doing this, we end up with 4 different categories of orphan receptors. Some of them, they have low or none or no expressed in human pancreatic islet. Some of them, like 21, they have a low expression. 35, they have a moderate expression, and 5 of them, they have high expression. When we correlate, if you look at the right figure here, when we correlate the expression from the microarray with the RNA sequence, blotting them together, we got really nice positive correlation between the 2 different technologies. So this is the first step. The second step as well, we even characterize these orphan receptor in the insulin cells, which is, as I mentioned before, it's the translation model that mostly we use when it comes to diabetes research. So this shows us that there are like 55 G protein coupled receptor common between INS and the human islets. Remember that the INS one is rat. So there are 55 common. There are 36 that mostly you will find it in the human, and there is 0 G protein coupled receptor that you can find it in the insulin cells, but not in the human islets. So what we have done next is to do a differential expression with all these 91 receptor in diabetic and non-diabetic. So we have done the test in 2 different ways. First of all, we compare diabetic islets with non-diabetic islets. Or, of course, when I say diabetic, this is the diagnosis that the physician has made and sent it to us. But in the lab, we have done our own category here. We use hyperglycemic versus normal glycemic. So this is the category that we have done in the lab. So doing these 2 different analyses, we end up with several genes that differentially express. Among them, we have found the GPR 183 and GPR 75. They are a kind of overlap between the 2 analyses. So we thought that this is something very interesting, that it shows us that these 2 genes are robust in terms of the finding. So doing more research on these 2, GPR 183 or 75, unfortunately we found that GPR 75 had been implicated previously with insulin secretion, but not the 183. Thereby we focus on our research on the 183 only. When it comes to the 183, you can see it here in the microarray that it's expressed there. Of course, the expression is not that high. It shows that it's in the lower percentile in the RNA-seq. However, doing immune staining, we were able to show that the GPR 183 is expressed in the human islets and mainly co-localized with insulin secreted cells, but not, for example, the alpha cells. As well, we look at the GPR 183 in different metabolic tissue like fat, pancreatic islets, liver, and muscle. And in all of these tissue, GPR was expressed there. So that was the important startup. So what is GPR 183? It's not that much known about it, but we can say a few words about it, that it's Epstein by virus-induced gene 2, that it's highly expressed in the spleen. It's required for human immune response, including B cell migration and T cell dependent antibody production. As well, it has been shown that there are metabolites called 7-alpha-25 dihydroxycholesterol work as a potent agonist for this GPR 183. So we start up first to look the role of GPR 183 in INS1 cells by knocking down this gene and look whether knocking down GPR 183 will influence the insulin secretion in the cell line. So we, if you look at the figure A here, you will see the silencing efficiency after silencing these 2 genes. We have used GPR 183 and 75. We have used 75 here as a control since it has been indicated. So we thought, okay, let's keep it on and use it as a control. So the efficiency of the silencing, as you can see, almost 80% of the genes on these INS1 cells. And when you look at the insulin secretion here, you will find that knocking down GPR 183 in INS1 resulted in a significant reduction of the insulin secretion compared to the negative control. So next we examine whether the silencing will influence the insulin production. Unfortunately, although we find a sort of reduction, but it was not significant. More we look at the consequences of the GPR silencing on beta cell function genes such as insulin, of course, gene, PDX, and MAF. These are very important regulator for beta cell function. So we, as you can appreciate from the figure, that all of them should to be influenced by significantly down regulated. So we have a finding here. We thought, okay, since the 7-alpha-25 dihydroxycholesterol has been proposed as agonist, so why not to try? So fortunately, as you can see here from figure A and B, when we use different dose from the agonist 7-alpha-25, 0, 10, and 100, 1000, we found that there is a sort of insulin potentiation for insulin secretion in human pancreatic islets in a dose-dependent manner. So the more you give 7-alpha, the more the human islets will secrete insulin. As well, we found that these agonists as well protect the cells by increasing cell viability if we culture these cells in high glucose medium. Knowing that high glucose medium, if you culture your cells, that will create a sort of apoptotic environment for these islets. But what we see here, that the 7-alpha protect these cells in high glucose culture medium. So we were very happy about this data. I don't want to talk about these two figures here because of the time, but maybe I can move for the next. So the next, we tried to study different type of mechanism for the GPR 183 by looking or investigating whether this gene or this agonist influence cell proliferation or cell cycle in the NS1 cells. So doing thymidine incorporation here in different dose of the 7-alpha dihydroxycholesterol, we found that a significant cell proliferation in a dose-dependent manner, as well as we doing cell cycle analysis for the NS1 cells culture and 7-alpha-25 dihydroxycholesterol, we found significant increase in the S phase and the G2M phase, but not on the G0 or G1 cells, which is in a way fit to the cell proliferation data. Lastly, we look at the effect of the 7-alpha-25 dihydroxycholesterol on certain beta cell function gene, such as, of course, insulin gene, PDX, GLUT2, insulin receptor, and glucose kinase. So these are, again, a very important genes in the beta cell and when it comes to its function. So what we found that the 7-alpha influenced the insulin gene, it influenced PDX, it influenced the insulin receptor, but not GCK or GLUT2. So by this, we conclude in our data that this study shows a comprehensive profile for all the orphan G protein covered receptor expression in human pancreatic islets and in NS1 cells. As well, we identify the GPR 183 is a novel gene for type 2 diabetes. We showed that 7-alpha-25 dihydroxycholesterol potentiate insulin secretion, reduce glucose toxicity, and increase cell proliferation. As well, important thing here that we show for the first time that show a novel mechanism for cholesterol metabolites in insulin secretion. And this is very important because we were the first to show that cholesterol, because it has been thought that cholesterol is a bad boy, but here we show no, it has some good effect. But this we can say finally that we believe that GPR 183 could be an attractive drug therapy for pancreatic beta cell function. And lastly, of course, I need to acknowledge all the people that share with me the work in this study. Of course, Al Jalila Foundation for their support. Of course, our colleagues at Lund University, Sweden, for their support and for data that we have got from them. In Sharjah, of course, this is my lab here. So this is our co-worker that shared the work with me. I would like to acknowledge all the people in the Diabetes and Metabolic Research Group and the Iron Biology Research Group. Finally, of course, this work had been supported by a grant, of course, from Sharjah University. And thank you very much. I hope I'm on time.
Thank you very much, Dr. Jalal. It was a very interesting presentation. Now, Dr. Ihsan will present.
Sharing.
Dr. Jalal, if you can please stop sharing. Okay,
Yes.
Thank you.
Okay, can you hear me? Have we got voice? Hi, this is, how's the sound quality? Is it all right?
Yes, we can hear you loud and clear.
Okay, perfect. Okay. Good afternoon. I'm Dr. Karimiani. I'm the consultant medical geneticist, clinician. And I'm talking about different genetic diagnostics and the opportunities and the challenges that we face during our clinics and seeing patients. There are a few troubles that I came across finding these kind of challenges. So first of all, we're thinking about in the clinic, and the families came to us. We think about that why genetics is so important, and especially for Middle East families with consanguineous background and first cousin marriages and stuff like that, and why the Mendelian disorders and the diagnosis of them is so critical at some point. So the exact diagnosis of patients is very important, that we have to do the genetic tests. Every patient and every family wants to know the cause. So in the genetic counseling, they're asking why that patient has got the epilepsy or mentally challenged or intellectual disability. And we see, and we're going to counsel them and talk to them. And that is very important. And a growing number of conditions are treatable now. So the genetic test has got a significant impact on the treatment of patients, such as different disorders, such as epilepsy, autism, and the personalized medicine precision approach in medical conditions that came to us, that pushes us to do genetic tests. And the management and the prognosis, we can talk to the family that how's the prognosis and how's the management, and also the genetic counseling and family planning and prenatal diagnosis and the risk of the recurrence. So that is why the genetic is so important. But there are some challenges, some problems over these kind of tests. So on the problem and the challenges is within undiagnosed cases. We've got undiagnosed cases that, despite the problem that we can see during the clinical counseling and the clinic that we see patients, that we see that the patients have perform different type of tests. They're still undiagnosed. Tests are negative. We know that some genetic elements are there, but we can't find them. So that came into across that we have to find a solution. So the center or the company who wants to deal with undiagnosed cases needs to be clinically orientated and should got an access to big databases and good knowledge of the population genetics, like founder effects and variants of unknown significance, and the populations such as Middle East or something like that, and further categories and access to further research tests such as gene mapping, whole genome sequencing, whole mitochondrial disorders, sequencing, and be able to reanalyze the big data. That is the key. That is the importance that we have to be able to reanalyze the big data using artificial intelligence, either artificial intelligence or the bioinformatician and big team that can use the AI-based computer vision approaches. Okay, and at the end, we have to be able to publish novel genes and report it to the family. Okay, so OMIM, everyone knows what is OMIM. OMIM is now and done throughout the history. The primary mission of the OMIM has been to collect the knowledge of the human gene and the genetic disorder. And currently there are over 24,000 entries and describing over 16,000 genes and 8,000 different phenotypes. But however, many genes are still unknown, and that is a challenge. So what we should have to do for next generation variant filtering. So the patient or the family come to the clinic, they're going to do the diagnostic services and the diagnostic test in trio or in solo. And we're going to do exome or genome sequencing, different companies are there, such as Origins that we have established quite recently. And if negative, we have to be able to reanalyze the data, having access to the big databases, and we have to know novel genes, even unpublished genes, using artificial intelligence, even different tools. All right, we can find the candidate genes, clinical review, the mutation analysis in the family, to match with and find other families as well, and then to perform the functional studies and report it report it or to publish as a joint publication. Okay, and then again, the patient, we can recall the family after even may take ages or many years, recall the family, and that we found a solution and we found a diagnosis. They're around. We know that the human genome, not all of the gene has been actively studied. Okay, we can give in the clinic and by the analysis of the human genome, we can give the clue to different universities that what we are looking for. Okay, there are about 20,000 human protein coding genes are there, but recent studies suggested that scientists actively study about only 5,000 or 6,000 of them. Okay, so about per year, about 300 new phenotypes are being published every year, and about 16,000 genes and 8,600 different phenotypes. But there are many genes are still undiagnosed. Every year, 300 different genes will be getting published. But we need to increase this, and to increase the diagnostic yield of the different diagnostic centers by having access or reanalysis perfectly the raw data of unknown and undiagnosed cases. Okay, so the value of data sharing is that important. This process can be greatly facilitated by connecting with other clinicians. We have to know each other. Patients and variants in the same candidate genes, we can find similar cases, similar phenotypes, similar mutation genes, and then doing functional studies and to publish that and to solve the problem. Okay, basic scientists and special expertise in the functional studies, such as zebrafish in mouse models, and with defects in the genes. They can knock down the genes, and they can see what happens in the human phenotype that can match with the clinic of those patients came to us, has been categorized as undiagnosed cases. Okay, so the final diagnosis may take even several years. I've got patients from 5 years back or even more than that. We record them. Okay, now the problem is solved. The papers are published, and we use your patients in order with all the ethical consent and stuff like that to publish this paper, and then we can talk to them after that. Okay, so I'll give you a few examples such as new genes such as that we've published quite recently. I have patients and with hereditary spastic paraplegia, some spasticity, and so exome negative was not conclusive at the first place. Okay, so homozygosity mapping was performed, and common locus was opted, and RNF 170, a new gene, and has not been reported for the HSP in the past. Okay, and then we performed the functional studies, and then functional studies on the zebrafish has got an effect on the brain of the zebrafish, and then we published that paper, and then recall the family, give them the diagnostic report, and even we've performed the PND, prenatal diagnosis for the next pregnancy. Okay. Or another novel gene called UGP2 in the brain leads to severe epileptic encephalopathy. Okay, you can see that there are 13 different families who are getting together, we find each other, same scenario, same clinical presentation, epileptic encephalopathy, which is refractory to different treatment, and all of patients being mentally challenged and with intellectual disability. And we've done the gene expression analysis, and the area was quite conserved. And so the final diagnosis was a new type of the epileptic encephalopathy. Okay, and also another gene called ADP or HL2. So these are all patients that have been unknown or undiagnosed for many years. Okay, and so 6 different families. All of this gene, by the mutation in this gene, has got an effect on the brain, on the back of the brain called cerebellum, and it causes pontocerebellar hyperplasia, and all of them has been diagnosed with the ataxia. And the disease is called pediatric stress-induced epileptic ataxia. So when the patients, all of them, has been under stress, they had episode of ataxia called epileptic ataxia. Okay, attacks of epilepsy with ataxia. Okay, so how do we find each other? So how do these families find each other? Into the gene matcher? Around 7,000 genes have been submitted to the gene matcher so far from different countries. About 6,000 gene matches, 1,300 different genes. It has enabled collaborations with description of novel Mendelian phenotypes, novel Mendelian diseases such as HPTL, MFSD2A, or UGP2 that are shown in the papers. So we are quite expert to reanalyze and collaborate with different centers and find similar cases in different parts of the world. And the Origins wants to give this service to solve those undiagnosed cases with a big team of clinicians, bioinformaticians, and also artificial intelligence people, expert people. Okay, the molecular basis of at least 3,000 phenotypes is still unknown, and many more Mendelian phenotypes not been yet described. Okay, so the main aim. is to investigate the genetic cause of inherited conditions, origins test using personalized medicine in epilepsies, cancer, and other multifactorial disorders, in order to help clinicians to have targeted treatment in a manner of precision medicine, to increase the diagnostic yield of the genetic centers and genetic tests by performing the novel methodology of the origins in gene discovery using artificial intelligence in big data genomics. Okay, and the origins can improve the precise genetic evaluation for the health and the nutrition, and we have so far identified genes that underlie rare and ultra-rare inherited conditions, and the majority of them are in neurology. Okay, and the prospect for the future is artificial intelligence will be used to solve rare cases for Mendelian phenotypes, and it is significant. It will significantly help clinicians well for especially cases who are still undiscovered or undiagnosed, mainly genetic problems like recessive disorders. The process of large and complex genomic data sets and AI-based computer vision approaches and effective placement of the artificial intelligence medical application, particularly those utilizing human genetics. And next, we focus on evolving methods for specific tasks in clinical genomics, including variant calling, genome annotation, variant classification, and phenotype to genotype correspondence. Okay. Thank you very much, and I would appreciate the St. George's University of London for the grants and for doing exomes and all the functional studies on different patients, the University of London, and my friend, co-founder of the Origins, Dr. Ali Kiai. Okay, I'm with you now.
Thank you so much, Dr. Ihsan. We're going to move to the questions which were asked by some of our participants. We have a question for Dr. Jalal. What are some of the challenges that you faced while working on the identification process of the genes, especially the GPR183 gene?
Well, I don't know the question really. What does he or she mean by this? Is it in lab work or a logistic obstacle or what?
Generally, like the challenges that you faced.
Well, generally, the challenge is, of course, that you need to have a good database that you can start working on. I mean, I was fortunate because I built up this database myself when I was working in Sweden. So when I moved to Sharjah, I moved it with me, but it's already published, so anyone can get access to it and do the same research as I have done. You need to have a good lab here to validate anything. You have to be careful what kind of cell lines that you need to use, type of materials that you need to test. Of course, here in Sharjah, we are doing really good nowadays because mostly we have everything you dream about. Electron microscope, animal facility, state-of-art lab, a good metabolomic facility, fax facility, everything. Now, I mean, I would say in the last 6-7 years, University of Sharjah has moved very quickly. So just bring your idea, and then you are welcome to establish it here. Or if you have an idea about it, of course, you could coordinate with someone or collaborate with someone to do it.
Wow, okay. Thank you. So the second question, I think it's for Dr. Ihsan. What was the process of the genetic testing like? For example, you presented the case for the severe epileptic encephalopathology patients, for the young patients that you had. What was the process of the genetic testing and how was it discovered?
Okay, I'll give you an introduction about human genome. Okay, so we've got roughly about 25,000 different genes, and if we've got So humans are different to each other by not by 1% human genome. So these dots that we are different to each other called variants. Okay, there are five. We've got five classifications of the variants: class 1, 2, 3, and 4, and 5. That's why we are different to each other. Okay, so classification of the variants. Class 1 is called pathogenic variants. We tend not to use mutation anymore. We use variants. Okay, pathogenic variants, likely pathogenic variants, variants of unknown significance, likely benign and benign. Okay, so you have to know how to filter those variants by sequencing. When we see doing the genome or exome sequencing, after filtering those, it's going to give us the variants, those dots that we are different to each other. Okay, but one of them, or at least one or two of them, are pathogenic variant that may have caused that problem, that epileptic encephalopathy, or other problems like mental retardations, or blindness, or deafness, or whatever. Okay, you have to find a needle in the haystack. Okay, sometimes it's very difficult because we do not know when the needle is in a bad gene, but we do not know the function of that gene that may cause hearing loss or the blindness. Okay, because it's not ethical to knock down that gene on the human being. Okay, it's not ethical. We have to find those patients who've got that variant in that particular gene, and not only one family, different families. These are ultra-rare disorders, and we have to match with other type of the families or other families with the same phenotype. We're going to do the perfect analysis and meticulous clinical assessment on the MRI or the clinical presentation or the type of the epilepsy or whatever. And if they match clinically with each other, and then bang, we're going to publish that, and we're going to do the function. Yeah, and also on top of that, if the functional studies matches with the clinical presentation, that is the key. Okay, and that is called gene discovery. And then everyone knows that in the world that if they've got a variant, pathogenic variant, or the mutation in that particular gene, it can be diagnostic. Okay. So that problem solved. Okay, I think there are 20 or 30% of the genes in human genome is still undiagnosed, still undiscovered. Okay.
Okay, thank you for answering the question. Dr. Ihsan, do you have currently a genetic lab testing in the UAE, or are you based in London?
Yeah, we're planning to have a genetic diagnostic or research in Sharjah Park, Science Park. Okay, hopefully in a couple of months or something. Okay.
Okay, so I'm just going through the questions now. So we have a question. Is there research or findings regarding having genome as memory storing certain human activities or parameters resulting from the activities? I'm not sure.
I think this is for maybe Dr. Ihsan, but I'm not sure I understand the question carefully. Dr. Ihsan, can you see Ihsan?
Can you repeat the question, please, for me?
Is there research or findings regarding having genome as memory storing for certain human activities or performance resulting from the activities?
Yeah, as I said, if I'm not mistaken, your specification or the variation between people such as sport, you know that some ethnical background, people with different ethnical background, they're more talented in some certain type of sports. For example, people in Jamaica, and they're good in running. People they've got a European background, they're good in swimming, and so these are nothing but the variations and those Previously, we call it a SNP, polymorphism, but we tend not to use those terms anymore. So the variants, those variants make people different. So the talents can be different. Your future can be different. So that these are can be affected on the environment as well. But the genetic is one of the factors. Some people in some families, some problems runs in some families, like diabetes, as Dr. Jalali was talking about that. So some genetic elements are there, but we have to find that, and we can counsel patients. And one of the another important factor is nutrition. Okay, nutrition is not only calorie based. So people who get on the diet, certain diets. So we cannot give same diet to different people because they are genetically different. Okay, it's not only calorie, but also your SNPs or your variants in the body. So we have to, that diet needs to be personalized based on your genetic information. Okay, the way and other things like sport and other stuff like that. Okay, these are, and genetic is not everything. I'm not saying that everything depends on your genetic information, but genetic is one of the important factors.
Thank you.
Okay.
Yeah. So we have a question, final one. I think it can be answered by both Dr. Jalal and Dr. Ihsan. How can the genome discovery work or help during the COVID-19 pandemic?
Well, yeah, that's a good question. Well, in fact, we have a sort of gene discovery when it comes to COVID-19 and diabetes. Remember what happened during the COVID-19, that diabetic patients are really suffering a lot compared to non-diabetic when it comes to COVID-19 infection. So we were trying to understand why. Is there a sort of link between COVID-19 and diabetes? Is there any sort of link between, for example, the lung tissue and pancreatic islet tissue? We thought there is a molecular link, and we have, if he's interested or she interested, she can go to bioRxiv and look at our study. We believe that there is a gene, like a link between the lung and the islets. We call it XO6. So, of course, this has been a prediction based on bioinformatics data. And at the moment, we are trying to validate our thought in vivo and in vitro. Hopefully we are right on this regard, but just give us some time and we will really answer you in a comprehensive way. But yes,
Thank you.
to answer, yes, there is a sort of link, definitely.
Thank you. Dr. Ihsan, do you have anything to add?
Yeah, not particularly, but the host and genome and the variants of in people's genome is very important, such as the signaling pathway of interleukin-1 has been very important. Different genes such as UBE2V1, as far as I remember, they've done the human genome sequencing in different populations in Chinese people, and they found out that there are certain genes in the immunity of people is important. Some people are susceptible to COVID-19, some people are more resistant, and so that is nothing but genes sometimes, and the discovery of genes. So we've got two different elements. As you see, the host genome of people, humans, and genome of the virus. Okay, so there is a the two different type of variants, two different genomes that make patients susceptible to the disease, or make the viruses on the other hand more virulent. the gene discovery can help this. And of course, if in the future on the treatment, and when they're going to do immunotherapy or gene therapy in the future that targets those bad genes that can be expressed in the disease that can help. It's not the field of my expertise, but something generally gene discovery and the host and the human genome that can be affected by the COVID-19. Okay.
I agree with Dr. Hassan as well that yeah, there is a sort of genetic variance that might tell you why this person has really complicated situation when it comes to COVID-19 and why that person is not. So it might be again to genetic variance. Yeah, I agree with Dr. Hassan.
Okay.
Thank you both. I think that would be the end of our Q&A session, and we can wrap up. Thanks again to both of our speakers, Dr. Hassan and Dr. Jalal. It was an honor to have you on board for our webinars, and to our participants as well. Thank you.
Thank you very much. Thank you. Thank you, Dr. Hassan.
Okay, thank you, Dr. Jalal. Okay, catch you later.
Thank you all. And the session is recorded on YouTube as well, if anyone wants to access it later.
Yep.
Okay, thank you very much.
Great.
Thank you. Bye.
Appreciate that. All right.
Yeah.